Hey everybody, PreMedMama here. Hope you're doing well today. It's an honor to have you here visiting my site and watching this video or listening to this video. I'm a mother as well as a pre-medical student. I'm so glad you're going to join me in studying for the MCAT. Today I'm in the Biology Review book by Kaplan. We're in Chapter 1 and we'll be looking at Section 1-5, Viruses and Subviral Particles. If you like what you see or hear today, please be sure to subscribe to my channel, leave a comment in the comment section, like the video, or share it with friends and family. All right, let's get started. Section 1-5, Viruses and Subviral Particles. At the beginning of this chapter, we discussed the cell theory and noted that viruses do not fit the definition of living things because they are acellular. Viruses may be as small as 20 nm or as large as 300 nm. For reference, prokaryotes are 1 to 10 and eukaryotes are about 10 times larger. Unlike eukaryotic cells, viruses lack organelles and a nucleus. Viral structure. Viruses are composed of genetic material, a protein coat, and sometimes an envelope containing lipids. The genetic information may be circular or linear, single or double-stranded, and composed of either DNA or RNA. The protein coat is known as a capsid. That's C-A-P-S-I-D, capsid. If an envelope is present, it will surround the capsid and is composed of phospholipids and virus-specific proteins. The envelope is very sensitive to heat, detergents, and desiccation, and thus enveloped viruses are easier to kill. On the other hand, viruses that do not have an envelope are more resistant to sterilization and are likely to persist on surfaces for an extended period of time. Because viruses cannot reproduce independently, they are considered obligate intracellular parasites or pathogens. Viruses must express and replicate genetic information within a host cell because they lack ribosomes to carry out protein synthesis. Right, guys, so viruses don't have ribosomes to carry out protein synthesis, so they can only replicate their genetic information by invading a host cell and taking over its machinery. Let's keep reading. After hijacking a cell's machinery, a virus will replicate and produce viral progeny or offspring called virions, which can be released to infect additional cells. So virions are like the children, if you will, the offspring, the progeny of a virus. Bacteriophages are viruses that specifically target bacteria. If you've listened to my previous videos or read this book, the first part of the chapter, you know that we learned a little bit about bacteriophages already. So bacteriophages do not actually enter bacteria, rather they simply inject their genetic material, kind of like a syringe or needle, leaving the remaining structure outside the infected cell. In addition to a capsid, bacteriophages contain a tail sheath and tail fibers, and the tail sheath can act like a syringe, injecting genetic material into the bacterium. The tail fibers help the bacteriophage to recognize and connect to the correct host cell. And I will include some links at the bottom of this video for additional images and videos to supplement your understanding of this concept. All right, moving into viral genomes. Viral genomes come in a variety of shapes and sizes. Some are made of only a few genes, while others have several hundred. In addition, viral genomes may be made of either DNA or RNA, And as we said earlier, they may be single or double-stranded in both cases. Single-stranded RNA viruses may be positive sense or negative sense. Positive sense implies that the genome may be directly translated to functional proteins by the ribosomes of the host cell, just like mRNA. mRNA means messenger RNA. Negative sense RNA viruses are a bit more complicated. These viruses require synthesis of an RNA strand complementary to the negative sense RNA strand, which can then be used as a template for protein synthesis. Negative sense RNA viruses must carry an RNA replicase in the virion to ensure that the complementary strand is synthesized. And I've got a great link at the bottom of this video, which links to Khan Academy, explaining this process a bit more. Retroviruses are enveloped single-stranded RNA viruses in the family Retroviridae. Usually, the virion contains two identical RNA molecules. These viruses carry an enzyme known as reverse transcriptase, which synthesizes DNA from single-stranded RNA. And as a side note, that process is called reverse transcription. The DNA then integrates into the host cell genome, where it is replicated and transcribed as if it were the host cell's own DNA. This is a clever mechanism because the integration of the genetic material into the host cell genome allows for the cell to be infected indefinitely. And the only way to cure the infection is to kill the infected cell itself. The human immunodeficiency virus, HIV, which you've probably heard of, is a retrovirus that utilizes this very life cycle, which is one of the characteristics that make HIV so difficult to treat. Real-world sidebar. In your book on page 27 says, both retroviruses and the process of transduction are under investigation as methods of gene therapy. It is theorized that retroviruses and transduction methods can deliver functional versions of missing or altered genes so that the correct proteins can be synthesized and certain disease states can be alleviated. Gene therapy is discussed in Chapter 6 of the MCAT Biochemistry Review book. And then on page 28 of the Kaplan book is an excellent image, figure 116, showing the life cycle of the HIV, human immunodeficiency virus. That's all for this video. Thanks for tuning in. Be sure to check out the links at the bottom, and please subscribe or like the video if it resonated for you. Thank you, and I believe in you.